control of mealybug. Thank you, Letodi.
Good afternoon, everyone. I'll be giving a brief presentation on the control of the obscure mealybug using entomopathogenic fungi in deciduous fruit orchards. So the obscure mealybug is a polyphagous agricultural insect pest that attacks multiple fruit trees. But in South Africa, it is a dominant pest of apples and pears. When the insect feeds, it excretes a substance known as honeydew. This honeydew acts as a substrate for black mold, and it contaminates the fruit produce. And in some instances, the mealybug also infests the produced fruit, and the presence of the mealybug and also the contamination of the fruit with the mold affects the marketability of the fruit and results in severe economic implication as the fruit gets rejected during export. So the main aim of our study was to evaluate the potential of local entomopathogenic fungal isolates as possible biological control agents of this mealybug. We did this by screening the pathogenicity of the South African EPF isolates against this mealybug in laboratory studies. And then we selected the most effective EPF isolates against the mealybug, and we mass-produced the conidia of this the EPF isolates. And then we developed a formulation process where we formulated the mass-produced conidia to maintain their longevity and pathogenicity against this mealybug. So for the screening bioassays, we found that two metarhizium isolates, Metarhizium pinghainsi and rubersi, were the most effective EPF isolates against the mealybug. Both isolates induced mortality of above 90% in the lab. So the two pictures here just shows the infected cadavers of the mealybug following exposure to the Metarhizium isolates. This diagram just shows the mass production process that we developed, where we were mass producing the conidia of the two most effective isolates against this mealybug. We used the blastospores of the EPF isolates and rice as the substrate to mass culture the conidia. We further formulated the isolates using different oils, mainly vegetable oils and mineral oils, and also diatomaceous earth to see if we can preserve the pathogenicity of the conidia over a certain period of time. And then the oils showed to be the most effective, or they showed to maintain the viability and pathogenicity of the mass-produced conidia relative to the diatomaceous earth. And we further tested, after a period of 2 months, tested the pathogenicity of the formulated conidia that was mass-produced, the one that we formulated using oils. And then they showed to induce mortality of the colonies of the mealybug, even when they were stored over a period of 2 months. So the oils showed to be effective carriers of the produced conidia of the Metarhizium isolates. So from our study, we showed that local Metarhizium isolates, mainly Metarhizium pinghainsi and rubersi, can be used as potential biological control agents of the obscure mealybug. And then these local isolates can also be easily mass cultured under lab conditions, and the mass-produced isolates can also be formulated to be used in the field to control the mealybug under field conditions. And also, the oil-formulated conidia, they do show that they can induce mortality of the insects when applied, even after storing them in the oils for a long period of time. And then our study has also broadened a range of possible biological control agents that can be used against this chemical-resistant insect in the field. Thank you.